In this video, we are going to discuss about the normal amount and production of cerebrospinal fluid, the circulation of cerebrospinal fluid, the ventricles of the brain, and ultimately the absorption of cerebrospinal fluid into the sagittal sinus. The entire cerebral cavity that has a capacity of about 1600 to 1700 ml that enclosing the brain and the spinal cord. About 150 ml of this capacity is occupied by cerebrospinal fluid and the remainder by the brain and the cord. The major function of the cerebrospinal fluid is to cushion the brain within its solid vault. CSF is formed at the rate of about 500 ml per day or 21 ml per hour, and the total volume of the CSF that remains inside the cerebrospinal fluid system is about 150 ml. So the daily production is about the 3 to 4 times than the total volume of the fluid that remains in the entire cerebrospinal fluid system. About 2/3 or more of this fluid originates as the secretion from the choroid plexus in the ventricles. So let's discuss about the ventricles of the brain. We have 4 ventricles, 2 lateral ventricle, 1 third ventricle and 1 fourth ventricle. 2 lateral ventricles are connected with the third ventricle via the foramen of Monro, and third ventricle lies in between the two thalami. The third ventricle then connects with the aqueduct of Sylvius to the fourth ventricle. Fourth ventricle lies anterior to the cerebellum and posterior to the pons and medulla. And the lower portion of the fourth ventricle is continuous with the central canal of the spinal cord. The point is called obex, from where the inferior portion of the fourth ventricle transitioned into the central canal of the spinal cord. And from the fourth ventricle, there are 2 lateral openings and 1 midline opening. And via these openings, the fourth ventricle is connected with the subarachnoid space. The lateral openings, L for lateral, L for Lusca. So the foramen of Lusca is situated on the lateral side of the fourth ventricles, which connect fourth ventricle to the subarachnoid space. And the midline, M for midline and M for Megendie. So foramen of Megendie is situated at the midline, which connects fourth ventricle to the subarachnoid space. So the foramen of Lusca and the foramen of Megendie are the main openings from which CSF is circulated from the fourth ventricle to the subarachnoid space, and little amount goes to the central canal of the spinal cord. The choroid plexus of the ventricles, mainly of the lateral ventricle, are the main source of CSF production, and the choroid plexus of the third and the fourth ventricle can also produce a CSF, but not that much amount. The other source of CSF production are secretion by the ependymal surface of all the ventricles and by the arachnoidal membranes, and a small amount come from the brain through the perivascular space that surround the blood vessels passing through the brain. So the CSF production has 3 main areas. One is the choroid plexus of the ventricles, number 2 is the ependymal surface of the ventricles, and third is the perivascular space that surrounds the blood vessels of the brain. So the fluid secreted in the lateral ventricles passes first into the third ventricle. There some amount of the fluid get added, then from the third ventricle via the aqueduct of Sylvius it comes to the fourth ventricle, where a still another amount of fluid is added. Finally, the fluid passes out of the fourth ventricle through the 3 openings, 2 lateral opening, 1 in the midline opening. The lateral openings are foramina of Lusca and the midline opening is the foramen of Megendie. Then from the fourth ventricle via these 3 small openings, the CSF enters into the cisterna of Magna. The cisterna of Magna is a fluid space that lies behind the medulla and beneath the cerebellum. The cisterna magna is also known as cerebromedullary cistern. It is the largest subarachnoid cistern in the brain. The brain cisterns, these are interconnected fluid space. These are not a closed space. The cisterna magna is continuous with the subarachnoid space that surrounds the entire brain and the spinal cord. Almost all the CSF then flows upwards from the cisterna magna through the subarachnoid spaces surrounding the cerebrum, and from here the fluid flows into and through the multiple arachnoidal villi that project into the large sagittal venous plexus and the other venous sinuses of the cerebrum. Then any extra fluid empties into the venous blood through pores of this villi. Secretion by the choroid plexus. The choroid plexus is a cauliflower-like growth of blood vessels covered by a thin layer of epithelial cell. These plexus project into the temporal horn of each lateral ventricle and the posterior portion of the third ventricle and the roof of the fourth ventricle. Arteries like the anterior choroidal artery and the branches of posterior cerebral artery, along with their corresponding veins, they form a rich vascular network inside the brain ventricular system. While entering into the ventricular system, pyometer also enters along with the vessels, as the pyometer is loosely attached here. So the vascular network enters into the ventricular system along with the pyometer covering. This is called the telachoroidy. The ventricles are lined by ependymal cells. When the ependymal lining covers the telachoroidy, it forms the choroid plexus. So choroid plexus project into the temporal horn of lateral ventricles, posterior portion of the third ventricle and the roof of the fourth ventricle. Secretion of fluid into the ventricles by the choroid plexus depends mainly on the active transport of sodium ions through the epithelial cells lining outside of the plexus. Choroid plexus is a projection of blood vessels that covered by a thin epithelial cells. The active transport of sodium through these epithelial cells is the main determinant for the secretion of fluid into the ventricle. As the sodium ions comes inside, it also attracts the chloride ion, as the chloride ion is the negatively charged. So the sodium and the chloride both increases the osmotic gradient, which then causes almost immediate osmosis of the water through the membrane, thus providing the fluid of the secretion. So the sodium and chloride increases the osmolarity, which attracts water via the osmosis. Thus via this, it provides the fluid for the secretion. Less important transport process moves small amount of the glucose into the CSF and both potassium and the bicarbonate ion out of the CSF into the capillaries. Therefore, the resultant characteristics of the CSF fluid becomes the following: osmotic pressure approximately equal to that of the plasma, sodium ion concentration also approximately equal to that of plasma, chloride ion about 15% greater than the plasma, potassium ions approximately 40% less than the plasma, and glucose about the 30% less than the plasma. Cerebrospinal fluid gets absorbed at the superior sagittal sinus via the arachnoid villi. Arachnoid villi are microscopic finger-like inward projection of the arachnoidal membrane through the walls and into the venous sinuses. At the anterior portion of the sagittal plane, the layers of the dura get separated, forming a triangular area, and that area is covered with endothelial layers, just like forming a capillary, or that is called the superior sagittal sinus, inside which there is some finger-like projections from the subarachnoid space occurs, forming the subarachnoid villi. The clustering of these villi form macroscopic structure called the arachnoidal granules that can be seen protruding into the sinuses. From here, the CSF get absorbed into the superior sagittal sinus via this subarachnoid villi. The CSF from the subarachnoid space only goes to the superior sagittal sinus via this subarachnoid villi, but the reverse flow of the blood from the sagittal sinus into the subarachnoid space is not possible. The epithelial cells covering the villi have been shown by the electron microscope to have vascular passage directly through the bodies of the cell, large enough to allow relatively free flow of cerebrospinal fluid, dissolved protein molecules, even particles as large as red and the white blood cell into the venous blood. In summary, the CSF is produced by choroid plexus of the ventricles, mainly the lateral ventricles, but some amount is produced by the third ventricle and the fourth ventricle as well. Some amount of the CSF is also produced by the ependymal cell linings of the ventricles and the perivascular space also. So the CSF, which is produced at the lateral ventricle, comes to the third ventricle via the foramen of Monro, and where the some CSF gets added up, that is produced at the third ventricle. So the total CSF from the lateral ventricle and some amount from the third ventricles comes to the fourth ventricle via the aqueduct of Sylvius. And in the fourth ventricle also there is some secretion of CSF. The maximum amount of the CSF of the fourth ventricle goes to the subarachnoid space via the foramen of Lusca at the lateral side and the foramen of Magendie at the midline, and a little bit also goes to the central canal of the spinal cord. At the subarachnoid space, the CSF moves around the cerebrum and the spinal cord as well, and then moves upwards to be absorbed at the superior sagittal sinus via arachnoid villi.